Hello friends, welcome to SAM Biology class. This is Subrat Kumar Mahanti. Today we shall discuss about crossover interference and coincidence. Crossing over is the interchange of reciprocal segments between the non-sister chromatids of homologous chromosomes. It is observed in many eukaryotic organisms that an existing crossover event inhibits the occurrence of further crossover events in the nearby region of the chromosome. This inhibition of further crossover events is due to interference. Crossover interference was discovered by Alfred Sturtevant and Hermann Joseph Muller in 1911 in Drosophila melanogaster. To understand what is interference, take the example of wheat in which 3 recessive mutations are linked on chromosome 5. These mutations are for whites and seedlings, for purple aleurone and for brown midrib. Now let us draw the linkage map showing the relative distance between these 3 genes. This is for V and this is PR for purple aleurone, and last one is for brown midrib. The distance between V and PR is 22.3 map unit, and that between PR and BM is 43.4 map unit. Assuming that there is no interference between crossing over, we can calculate the expected frequency of double crossover. The expected frequency is equal to 20.3 divided by 100 into 43.4 divided by 100, and this is equal to 0.223 into 0.434. Multiplying these two will get 0.097. So this is the expected frequency of double crossover. This is the frequency we are getting when there is no interference between crossing over, and this is equal to 9.7%. So this is the expected percentage of double crossover. Then what is the observed percentage? The observed percentage of double crossover is equal to 7.8%. So in this case, the observed percentage is less than the expected percentage. This difference between observed value and the expected value is due to interference. It is an example of positive interference because of which fewer than expected double crossover occur. Interference varies from chromosome to chromosome. It also varies even within the chromosome from one region to another. It is maximum near the centromere and at the chromosome tips. Interference increases when the distance between the genes decreases, and when the distance between the genes increases, interference decreases. To measure interference, we use another term known as coefficient of coincidence C. It is the inverse measure of interference and is expressed as the ratio of observed frequency of double crossover and its expected frequency. So in this example, C is equal to 0.078 divided by 0.097, and this is equal to 0.804. So this is the coefficient of coincidence. After determining this, Interference can be calculated by a simple equation I is equal to 1 minus C. So in this example, I is equal to 1 minus 0.804. This is equal to 0.196. This is the interference. It means 19.6% fewer than expected double crossover occur. It is an example of positive interference which is seen in mostly eukaryotic organisms. Interference is the complement of coincidence. Thus, I plus C is equal to 1. So, when interference increases, coincidence decrease, and when it decreases, coincidence increase. When we say that interference is complete, then I is equal to 1, and in this case coincidence is equal to 0. But when interference is 0, then coincidence is equal to 1. Interference is of two different types, positive and negative interference. Positive interference is seen in mostly eukaryotic organisms, and in this case fewer than expected double crossover occur. As a result, the interference is a positive number. For example, in Caenorhabditis elegans, a single crossover event per chromosome is observed. And also in Drosophila, interference is complete within a span of 10 Mach units. Second type is negative interference, in which an existing crossover promotes the occurrence of further crossover events in the nearby region of the chromosome. And in this case, interference is a negative number, and here more double crossover than expected occur. So friends, this is all about interference and coincidence. For more updates, please subscribe my YouTube channel, Sam Biology Class.